Hi. So this screencast is going to be about your humoral immunity response, or what happens when B cells are first exposed to an antigen and what leads to the production of antibodies. So it's going to start with antigen challenge, which is going to select the B cell that is needed. That B cell is going to proliferate into what we call a clone, a number of identical cells that all produce the same antibody. The members of this clone are going to then differentiate into plasma cells that secrete antibody and memory cells that hang around and wait for the next infection. So let's get started. First thing you need to remember is that each one of your B cells is unique. It produces a unique antibody unlike any of the other antibodies that you have. This is why the process of selection is so important. It starts with antigen challenge. I've skipped, let me go back one. Antigen challenge is the first time that you encounter an antigen. At this point, your B cell is referred to as naive because it's never been exposed to this particular antigen before. So we're going to start with antigen challenge and then move through these three things that we've talked about already. Here you have your antigen. It is the first time your body has been exposed to it. So that antigen is going to bind to only specific antibodies. Here you have a B cell which happens to produce an antibody that can bind to this antigen. This lets this B cell know that of all of the B cells in the body, this one produces an antibody that can recognize this particular antigen. So your body needs lots more of this particular antibody as soon as possible. This stimulates the B cell to proliferate. So you go from one cell that produces a specific antibody to thousands of cells that all have the ability to produce that same antibody. This is what's called the clone, this large group of cells that all produce the same antibody. The members of this clone are going to have two different fates, or they're going to differentiate into two different types of cells. Most of the cells of this clone are going to differentiate or become plasma cells. The plasma cells secrete the antibodies. So those are these guys down here, which are actively secreting the antibody, and that antibody is going to be secreted into the bloodstream, circulate around the blood and the lymph, and it's going to do all of the things that antibodies do. Other members of that clone that don't become plasma cells are going to become memory cells. So these are cells that, as it says right here, are primed to respond to the same antigen. So they're already kind of trained up and ready to go. And what you need to remember is that even though this figure here shows just one memory cell, you would probably have a few thousand. So that having memory means not just having one cell that has seen this antigen before, but having probably 1,000 or more cells that all have the ability to produce that antigen, so that the second time you get exposed to that antigen, you have 1,000 cells waiting to start producing antibody instead of just one cell. So this is the whole process in one slide. It starts with antigen challenge, the first time you're exposed to an antigen. That antigen will bind to only specific antibodies. So the B cell that produces the antibody that recognizes that antigen is selected. It proliferates, undergoes mitosis many times to to produce a small army of cells that all produce the same antibody. Most members of that army will become plasma cells. Those plasma cells will secrete the antibody, and that antibody will go and inactivate or mark the pathogen for destruction. Some of the members stay behind as B cells, so that, again, the next time you get exposed to that, you have many B cells ready to differentiate into plasma cells and secrete large amounts of antibodies quickly. So that is antigen challenge, clonal selection, which leads to proliferation, differentiation, and these two different fates of the clones. All of that in under 6 minutes.